Good morning and welcome to today's MPG primer session.
We are very excited today for a talk that I think will help us all leap into the genomic future. Dr. Margaret Sunitha is going to share with us the advances she's made as really like an alpha tester of cross-cohort cloud computing using genomic data. So she is a research fellow in medicine at Massachusetts General Hospital and also here at the Broad Institute. Prior to that, she was at Bristol Myers Squibb's, and prior to that at the Institute for Stem Cell Biology and Regenerative Medicine. So you have a wide variety of expertise that I'm so excited to hear about how you brought to bear on this very important question of how to leverage the most out of our genomic data that's available. So thank you so much for joining us today. Is it okay, I forgot to ask you this, if you take questions during the talk?
Yeah.
Perfect. So if anyone's here, please feel free to raise your hand. And then anyone who's watching remotely will also monitor the Q&A. Thank you so much.
So I'm Margaret. I am a genomics/bioinformatics researcher. I work in Dr. Pradeep Natarajan's lab at MGH, and I'm affiliated to the Broad Institute. I would like to thank the MPG Primer organizers for inviting me and providing me with this opportunity to speak in this forum. The title of my talk is Cross-cohort analysis via cloud genomics. So in today's talk, I'm going to talk about our recent work where we have carried out cross-biobank analysis on the cloud environment. And here we have detailed the pipeline for carrying out cross-biobank GWAS on cloud, and also unraveled the current capabilities and future opportunities that are present in these research environments, which hosts the genomic data for the researchers. And multiple publications have pointed out the benefits of carrying out large-scale genomic analysis on the cloud. And this is because on the cloud, we have the right resource to securely host the genomic data, and we have the right tools and platforms to develop apps and pipelines which can be applied on the genomic data. It has the right compute environment to carry out large-scale genomic analysis, and also the storage for having both the primary genomic data as well as the secondary data that researchers generate during their analysis. So all of this is there in one place on the cloud. So cloud is good for large-scale genomic analysis. But it becomes complex when there are multiple such cloud platforms available for researchers. So for example, here, all of this genomic data is being hosted in the researcher workbench, which is powered by Terra. The UK Biobank genomic data is hosted in the research analysis platform, which is enabled by DNA Nexus. And NIH hosts its genomic data in different platforms, which is enabled by Anvil and Biodata Catalyst. So when these genomic data sits in different cloud environments, it becomes complicated for researchers to carry out cross-biobank analysis. So having said that the size of the genomic data is increasing day by day, the security around these sensitive information is also increasing day by day. So that is why researchers are presented with trusted research environments. So a trusted research environment, or a TRE, is a secure computing environment which provides researchers who have approval to, with the right tools and workflows to access and analyze these sensitive health data. So in this project, we have compared two such TREs. One is the All of Us Researcher Workbench, which hosts the All of Us genomic data. And the second is the UK Biobank Research Analysis Platform. So in this project, we have characterized the current advantages and limitations of these TREs in executing a cross-biobank analysis, and also by performing a genome-wide association and study in these two TREs. The phenotype of our interest was blood lipids. Let's get into the details of the work. So what did we do? We performed a cross-biobank genome-wide association analysis using both All of Us genomic data and the UK Biobank genomic data. So in the all of us, we used the 100,000 whole genome sequenced data, which is processed by GATK pipeline. And in the UK Biobank, we specifically used the 200,000 whole exome data processed with deep variant. There had been multiple releases of UK Biobank after this, but we specifically chose the 200K whole exome, and I will come to the reasoning in a couple of slides later. So the phenotype of interest was blood lipids. We included HDL cholesterol, LDL cholesterol, total cholesterol, and triglycerides in our study. So why are we doing this analysis? These are the two large cohorts which are available on TRE environments and cloud environments. And that is great for researchers, right? It opens up a lot of avenues to carry out cool research. But what happens if when someone wants to analyze both the cohorts together? So that is what we are exploring in this project. And though we use blood lipids as our phenotype of interest, this project is beyond discovery of lipids and more of a technical piece where we wanted to provide reusable codes and documentations for researchers to carry out analysis in these TREs, and also potentially identify policy improvements and technological improvements which can be used in the future in improving these TREs and as well as even emerging TREs. So the overall goal of this whole project is to make it easier for the future researchers to co-analyze these and emerging important data sets. So how did we do this analysis? We carried out GWAS in two different ways. One is the meta-analysis workflow and other is the pooled GWAS. So in the meta-analysis workflow, we carried out GWAS in two different places, in the UK Biobank RAP and the All of Us Researcher Workbench independently and as a separate workflow. Whereas in the pooled GWAS, we were able to use the 200K whole genome, whole exome data of UK Biobank, and that is why we used that release of the data, because that is the last release of UK Biobank data which can be used outside the RAP work environment. And so we carried out meta-analysis and we also carried out pooled analysis. So in the initial stages of the project, right, we invested a lot of time in understanding the policy constraints between these two TREs. Though they are all on the cloud environment, they do have slightly different policies. An example could be the data download and data dissemination policy that they have. Like the UK Biobank has the same rule for data download and data dissemination, whereas All of Us has a stricter rule for data dissemination when compared to the data download when compared to the data dissemination. So we had to work with these policy constraints to figure out the right way to develop a workflow through which we can analyze these two biobanks. And we also spent a lot of time to understand the functionality of the TREs. So these TREs are built on different platforms and they have different functionalities in terms of developing a workflow within them. So we spent some time in understanding the functionalities as well. So finally, after we had the meta-analysis and pooled GWAS results, we wanted to compare the scientific differences between these two methods and also compare the analyst productivity in carrying out GWAS in these two different ways. So, we project in a way that it can be easier and reciprocal and scalable for any future TREs when they could follow such protocol. On the meta-analysis workflow, so these are called as cloud plots. So you have a lot of arrows here. I just wanted to point out the main important points of this whole workflow. So this is the meta-analysis workflow where we carried out GWAS in the two different cloud environments separately, independently. And so in each of the cloud environment, for example, in the UK Biobank RAP, we prepared the lipid phenotypes, we prepared the genomic data file, and other auxiliary files that are required for GWAS, and we ran GWAS using Reginy. And exact same steps were carried out in the Olafers researcher workbench, and where we prepared the phenotypes and did the GWAS using Reginy here. So once we had the raw GWAS results, then we applied the data dissemination rule. And we used the Olafers data dissemination rule, which is the more stricter one, which says that any variant with an allele count less than 40 should not leave the trusted research environment. So we removed all the variants applying this rule equally on both the TREs. And then we aggregated the results which are okay to leave these TREs. So once we had those files, then we carried out a combined GWAS meta-analysis using Meta, and then created plots and summary statistics which are combined of these two cohorts. And this specific result is shareable results. So then this is the cloud plot for the pooled GWAS. And as you could just see here, that the number of steps that we had to carry out on the pooled GWAS is way lesser when compared to the meta-analysis. That was because we had the 200K whole exome data in the same cloud environment, and we did all the analysis in the Olafers researcher workbench. So the blue boxes that you see are the same steps as it was carried out in the meta-analysis, but we also had to carry out a couple of more steps to aggregate or integrate the UK Biobank and Olafers data together. So we prepared the UK Biobank lipid phenotype on the cloud environment in the Olafers workbench, and then merged it with the Olafers data. And similarly, we created UK Biobank Hale matrix and then merged it with the Olafers genomic data. So we again applied the same data dissemination rule, removed off the variants with allele count 40, and then aggregated the results to have the shareable files. And there could be a question of how did we mitigate the batch effects, right? Because here we are dealing with two different biobanks, sequenced in two different ways, processed by two different bioinformatics pipeline. And how did we address this question? So in order to answer to that question, we did adjust for the cohorts as an additional covariate in the model in the pooled analysis. And also both in the pooled analysis as well as in the meta-analysis, we did account for variants that were present in both the cohorts. So we removed off variants that came from only one cohort. So we had the set of variants that we filtered to the set of variants which were present from both the biobanks, and then carried out the rest of the analysis using that set of variants. So that is how we try to address the two cohort batch effects. Yes, please.
So what was the interface like? Like, did you use WDL or some sort of another SnakeMake or whatever language which was used? That's one. And two is how are you recording all these steps? In what format are they be recorded, as JSON format or whatever the format you're recording, so that somebody who can go and take a look at it, what exactly was done at what steps, not 2 was done before 3, or 2A, there was a 2A step in. And so all these, how are you tracking all these things? Thank you.
Thanks for this question. And I wanted to point out this through my talk. So we have used Jupyter notebooks with R and Python codes to carry out all these analyses. And to answer the second question of what is the exact steps of how we carried out, we have a complete documentation of all of this, and I can show you in the last section of my talk, where we have stepwise code which could be ran by a researcher at their end with all exact commands that were used to run this analysis. So it is like create PCs. What is the first step? Like, how did we create the phenotype? How did we create a cohort for lipids in the Olafers? How did we curate the lipids, normalize it? Then how did we create the PCs? Variant QC that we had implemented, every step is documented, and I can show you in the last section of this presentation. Yeah, so this is the overall workflow for meta-analysis and pooled analysis. So this is the two different workflows that we implemented. Here I wanted to show you some numbers. this workflow shows the exact set of variants that we filtered at each step. And I wanted to point out a few things here. So we started off with all the data from Olafers and UK Biobank and in the meta-analysis, and whereas in the pooled analysis, we merged them together and started off from that data. And I also wanted to point out that we filtered out variants that were present in both the cohorts to mitigate the batch effect. And this step was carried out right in the beginning in the pooled analysis. And then we went through different filtering steps. And in the meta-analysis, we did filter for variants that are present in both the cohorts at the last. So at the end, we found 454 significant biallelic exonic variants from metal and 445 significant biallelic exonic variants from the pooled GWAS. So having talked about the workflow, I wanted to spend some time to describe about the scientific differences that we found while comparing these two analyses. The first thing that we was to look for a lipid loci. Since we use lipids as our phenotypes and lipid is well characterized by multiple studies, we wanted to see if both meta-analysis and pooled analysis was able to identify all important lipid loci. And we were able to see that all the important loci like LDLR, PCSK9, ApoB were all present in our analysis, and the genome significant list, the set of genome significant variants from this analysis were already being demonstrated to be significantly associated with blood lipids. We compared our results with known lipid studies. So we, in our lab, we have a recent paper where we had used TopMed freeze 8 whole genome sequence data and studied blood lipids extensively. So we compared the results from this analysis to TopMed whole genome sequenced data set. And we also compared with Hindi et al.'s paper, which is whole exome data, where they have extensively studied lipids. So the graph that you see here is the x-axis is the concordance with the meta-analysis, and the y-axis is the concordance with the pooled analysis, where we have taken the effect estimates from the genome significant variants and compared it with the 4 different rates that we have studied and compared it with the 2 different published literature. In general, the concordance was pretty high, and there was a slight reduction while comparing the pooled analysis with the Hindi et al. Paper, but when you look into the R-square values, they were pretty high. So the concordance between this current study and the published literature was high. Also looked into the sheer number of variants that we were able to pull from these analyses. So we looked into the plot that you see here is comparing the number of variants that are present in pooled analysis, comparing to the minor allele frequency. So the first graph shows the minor allele frequency in the x-axis and the count of variants in the y-axis, which are present in the pooled GWAS, whereas the second plot shows the variants that are present in pooled GWAS, but not in the meta-analysis. So we see that many variants were a lower frequency variants. So the pooled GWAS was able to capture a lot of rarer variants when compared to the meta-analysis. And this was one main observation from this study. We also compared the ancestry specific variants. So we went and looked back at the 400 odd genome significant variants that came from both meta-analysis and pooled GWAS, and we were able to see 370 variants to be common between both meta-analysis and pooled GWAS. And here the plot just shows the percentage of those variants in terms of ancestry distribution captured through nomads populations, and 370 which were common. And in meta-analysis, there were 84 variants present only in the meta-analysis workflow, but not captured in the pooled GWAS. And there were 75 variants which were specific to the pooled GWAS and not present in the meta-analysis. So when we go back and check at their ancestry distribution, in pooled GWAS, those variants were much rarer And also they were present in specific ancestry like American and African ancestry more when compared to the meta-analysis. And when we look back at these variants, they had a better CAT score, rarer variants, had functional consequences, etc. And such variants were captured through the pooled analysis, but not through the meta-analysis. So what this tells us is that these two analytical approaches could yield very similar results, but they are not identical ones, which has very important implications while studying diverse variants from diverse populations. And that is one observation that we saw while comparing the scientific differences between two of those analysis methods.
Margaret, I wonder if you can comment at all. I mean, something, but it seems to me that the strategy for dealing with batch effects by requiring things be present in both could remove some variants that may be specific to a population that was captured by one of the data sets and not the other. I wonder how often in practice you anticipate that to be a challenge.
Yes, that is a challenge. So we were able to see such variants. Here I'm just showing about the lost variants in terms of allele counts, but not in here the plot does not talk about the ancestries, but we did see that like just because they are not present in one cohort, we had lost a lot of variants, but we wanted to keep it consistent between the cohort and wanted to develop the workflow which can be applied both. But that is one valid point here. So we also went to check for these variants that were lost at different stages. So 370 variants are significant in both the approaches. There are 84 variants that you see here that is significant only in the meta and 75 significant only in the pool. And that was because few of those variants did not meet this criteria of being present in both the cohorts. And they were also filtered at certain stage of the analysis where we applied certain QC metrics. And this plot shows like, we had placed a hard threshold for genome significance here. Few of those variants which were significant in one method versus the other were just below that significance threshold that we placed. So this is a plot that shows the -10 log p-values of those variants that are present, and the ones that are in pooled analysis but were not captured in the meta-analysis were just below the cutoff that we placed. So these are scenarios in which we have lost variants. And as she mentioned, it is very true that because all of us is a multi-ethnic cohort when compared to UK Biobank, ancestry-specific variants could have been in this cohort, which was not captured in UK Biobank, and the threshold that we placed could have removed them. But we wanted to have a filter that could be, where we could compare equivalent results from both the cohorts. Described the pipeline and talked about the scientific differences, but I wanted to show some highlights on the complexity and the cost for carrying out this analysis on the cloud environments. So first on complexity. So these are the two cloud plots that I talked about earlier. And here where the arrows are numbered in each of those plots, and an arrow means that it is a computational step which has an input and an output. So in meta-analysis, we had 40 such steps to be carried out, whereas in the pooled analysis, it was half the time as that of the meta-analysis, which shows that meta-analysis was complex when compared to the pooled analysis. And also think like what happens if we had another TRE to this, right? Like if we had 1 or 2 TREs here, it is going to linearly increase the number of steps and the complexity of the analysis. So in general, meta-analysis was more complex when compared to the pooled analysis. And the other place where we had difficulty is that while carrying out a phenotype modeling, for any phenotype of interest, like here we had used Snippets, but any phenotype for that matter, when these row level data are present in 2 different environments, when you wanted to try, harmonize the data adjust for something, normalize it for something, it becomes so difficult to have it in two different places and figure out outliers between them to carry out these cross-cohort analysis. It's like you eyeball it between these two monitors and figure out how to move around it. So that was one difficulty that we faced when we were doing meta-analysis, when we had the data in two different places. Next on the cost part, right? Cost is very important while carrying out analysis on cloud environments. And here I wanted to describe cost as cloud resource utilization and percent time. So when we cloud resource utilization, so it is good that these TREs host the bulk of the genomic data for the researcher. It is all available, and researchers are freed from the burden of having that whole genome data. But they also provide the genomic data in a particular format, right? And certain level of QC applied to that genomic data. But there are tools researchers could use for genome-wide association analysis may not ingest that particular format of genomic data. So there is a level in which where you need to apply some QC and then convert the data to a format that can be ingested by these tools. So in the current analysis that we carried out, we had most of the cost was incurred at the compute step, which involved HALE, where we had to convert the HALE metrics table to a BGen file or another file which can be ingested by Regini, like a software which could carry out genome-wide association analysis. So, but otherwise, the rest of the steps where we used R, Flink, or Regini was less cost consuming, and it was in the order of cents to few dollars. So, so that is one factor when we consider cost and carrying out analysis on the cloud. Yeah.
Sorry, could you clarify, so those steps that you did with HALE, and are you implying would have done those same steps before?
No, that's not my point. So what I said is like we had to convert the HALE matrix table to a BGen file for it to be ingested by Regini. So that conversion step, was the most cost-consuming part in this whole workflow. But if we had to carry out a GWAS inside HALE, that could have not been the case. But if a researcher wanted to apply another tool for GWAS, that conversion becomes mandate. So since we don't have that data available, so in the whole process, that was the most cost-consuming. Rest of it wasn't. That's what I meant.
Things hardly go as expected. How much time did you have to spend on finding errors, fixing them, and how much cost did it occur to you?
So that is the second point here. So I showed that pretty cloud plot, which looks logical of going one step to the other, but that was not the case when we did this analysis. In reality, we had to iterate multiple times to figure out the right QC metric and right methods to follow, right steps to follow. That incurred cost. So that is one factor that needs to be considered while carrying out analysis on the cloud. So, so the data harmonization and the quality control steps were iterative multiple times we had to carry out to get to that workflow. But good to our project, there is something to start off for the researchers, and I can show you like where you can find those resources. So, so that is the second point that I wanted to mention, that cost. This factor, considering this multiple iteration, should also be considered while talking about cost. The third point about cost is the person's time, right? So it also depends upon how much a person has the familiarity with the TRE, the functionality of the TRE, and the tools that are present in these TREs. So when researcher just starts to explore this, maybe they are not familiar with it, and doing things multiple times would definitely incur some cost. And that did happen to us also when we started this analysis. But in general, the person time for the meta-analysis was twice when compared to the pooled analysis. But as I mentioned, all the other factors needs to be considered when someone talks about the cost in the cloud genomics. So now I wanted to spend some time showing the current capabilities and future opportunities that are available on these two TREs. So I put three sections. First, here I'm going to talk about the data access and safeguard of the genomic data. So the existing capabilities in these TREs are, it does have a centrally funded copy of the genomic data. Both UK Biobank and the OLIFIS do have the genomic data that can be accessed by the researchers. But as an opportunity, we see that there could be some kind of temporary working file which can be placed outside the TREs, like a common phenotype file across biobanks or something like that, which can be kept outside the TREs, which researchers can access. That is one point. And the other point that we observed through our analysis is that here we are placing a very hard data dissemination rule, right? So any variant with allele count 40 is less than 40 is actually removed. This is okay right now when we are looking at common variant GWAS, but if there is interest in looking at the rare variants in these cohorts, then it becomes tricky. So there could be some engagement with the participants around the potential scientific value of having this kind of analysis carried out on the TRE. Could provide maybe an opportunity to get those set of variants for the analysis. That is one opportunity there. And one main thing is that these TREs can actually support mirroring of several data sets in a mutually trusted multi-data set TRE or a federated TRE, which can mirror certain kind of data sets, which would be useful for the researchers to carry out analysis instead of doing it in two different places and trying to merge them together. On the research support. Right now, there is an excellent researcher onboarding effort put up by both UK Biobank and OLIFIS. They have tutorials, videos that are available for researchers to understand these TREs, and that is good. As an opportunity, we also think that cross-cohort TRE analysis with examples of how to run each step could be documented. And through our project, we have attempted to provide such resource for the researchers. And this kind of workflows can be implemented in these TREs for future researchers. The last is the analysis infrastructure. So right now, we do have these TREs have the capability of hosting different tools, packages, like it has the ability to host, have Docker image containers or Jupyter notebooks. And it also in both of these TREs, there are workflows developed, which is pretty good, where a researcher can use an app to do step 1, step 2, step 3, combine them to all together. That is all available. And but as an opportunity, what we see is that they could also include some common vocabularies, which can be used between multiple TREs, because each TRE has their own functionality, own way in which their researchers have to code and access data. So maybe a common vocabulary between the TREs could be helpful. And the other important thing that we wanted to point out is to use standard analysis application interfaces that comes from like global alliances for genomics and health. So this could be one common platform or common way of representing or having workflows developed in these two TREs. So this is one opportunity that is available in these trusted research environments. In summary,
Sorry, one quick question. You just mentioned Docker images, but Docker images are static. So I was wondering, could you elaborate on that? Because when it comes to workflow, there is the cohesive and coupling. So how easy it is to decouple, and like you mentioned, maybe somebody have done all the heavy lifting and we just need to take from step 4, or we want to include, let's say somebody did not use seeker to filter, so we want to bring in seeker. So how easy it is to decouple, make it another workflow out of it, and use that for analysis?
Yeah, that is a good question. So right now, these TREs have a developed app which could go one after the other, but fitting in another method into it, I think WDL kind of programming languages help us to have these kind of applications to it. But again, as I see, like it is an area which is improving day by day, and there could be a lot of improvement. But right in this project, we did not use additional workflows like that. Here, we did apply standard Jupyter notebooks and Python codes here. So that is one important point to consider. See, I have talked about our work where we have a demonstrative project where we have developed workflow and a method to carry out cross-cohort analysis between UK Biobank and all of us data. And I've also shown some differences between these two analyses that we carried out, the meta-analysis and the pooled GWAS, and also talked about the utilities of TREs, the capabilities and opportunities of these TREs. Now, I wanted to talk a little bit more on the demonstrative project that I said. So we have a lot of resources available through this project, which would be really helpful for the researchers. Demo project in the Olafus researcher workbench. So when you go to, when a person logs into their researcher workbench in the Olafus cloud environment, and if you click these 3 bars here, you will get into the featured workspaces. And in the featured workspaces, there are tutorial workspace and demonstrative workspaces. And if you click the demonstrative projects, you have a lot of demo projects that is being exhibited in the Olafus researcher workbench. And these projects are basically, it provides you with the codes and the methods that they have applied for addressing each question. So we also have our demo project here, which is named as demo siloed analysis of Olafus and UK Biobank genomic data. So if you go click into this project, you will be presented with the analysis page, which has the Jupyter notebooks, which were exactly used for this analysis. To answer to your question, we have all these resources here. So we have also numbered them in the way that we had implemented. So as I mentioned here, this is the final output of this whole work, but this had multiple iterations to come to this level. And so here I wanted to point out that we have a resource to run these scripts, Jupyter notebooks in the background. So, so the way the whole workflow is written is that you create a virtual machine in the researcher workbench. For handling tail jobs, you need to have a data prop cluster. And for any other regular jobs, you need to have a standard VM. So you create a VM and then you run all your scripts. Once you're done with that set, when you get the output file, you copy the output file to the Google Cloud bucket. So then you're done with the VM. Sometimes the VM crashes, gets deleted in these cloud environments, and you end up losing all the data that you stored there. So the way we have implemented this workflow is that you copy it, every step is copied with a date and timestamp to the bucket, so that you will know which set of analysis was carried out at what day and time. So that is how this workflow is being developed. So there is a run notebook in the background Jupyter file. So you just need to provide the notebook that you wanted to run, and it runs that notebook on the background and then provides you with the output with a date and timestamp here. So it is a Jupyter notebook file, so where it handles both R and Python commands. So each command is run and the output is also saved there, so you know which step has been finished, and if it errors out, you will get the error and it will be date and timestamp. So you will know which run it is.
So are there some pointers which you can give about how to request a resource? Like you mentioned VM, they can fade. Sometimes they may run out of memory. So was there some initial thinking about if you have that much of data, how much you should start with so that you have less failures because of shared memory issues?
Exactly. So in these notebooks, we also have a setup or, general information section where we have provided that what would be the optimal number of codes or what is the number of workers you need to have for a Hale job. We have provided that information. And we also have provided an estimate of how much it costed for this the bigger chunk of the, section where we use genomic data. So we have provided that information there. So, and yeah, as I mentioned, it is ordered, each step can be implemented and it can be run by yourself. So you just need to, duplicate this featured workspace to your workbench and you can start running it at your end or also use those snippets, code snippets that are available through this method inside your project. I just wanted to show a very brief set of snippets from the Jupyter notebooks. So we have this right matrix table section where we provide you also with the time that it took to create these bgen files from Hale matrix. And this, just to point out here, we have the whole genome data from all of us, but it was mapped to the UK Biobank exome capture region. So the size of the data is way lesser when compared to the whole genome data that you will have from all of us separately. So we have that step documented. We also have this setup documented. Like, for example, you create a VM, you just pull out some tools from their repositories, install the tool, run the analysis, and maybe if the VM deletes, it's okay. So we have that set of command where you can pull the tool from wherever you want, like Link or Reginy, and then execute it at that stage and start your analysis. We have the setup page also being documented. We have Reginy step 1, step 2, variant QC, PC aggregations, etc. available. And we also have the code to aggregate the results together, apply the data dissemination rule to it, and then create these aggregate files which can be taken out of the TREs. And we also have scripts to plot the data, a Manhattan plot, a QQ plot, or even plots to look into the phenotype file. So all of that is all there in one place. So I would urge you to look into these resources. Also have a GitHub repository with the current codes for both meta-analysis and the pool GWAS. So that is also available, and it is under the All of Us GitHub repository named as UK Biobank Cross Demo Project. So these were the resources that came out from this project, and we think that it would be useful for the researchers. So with this, I would like to acknowledge the project team. As you could see here, the project team is extremely diverse. There are folks from NIH, All of Us, UK Biobank, Vanderbilt Medical Center, MGH, Broad, and Verily Life Sciences. I would like to thank them all for the excellent collaboration, and a special thanks to Nicole from Verily Life Sciences. She has contributed a lot to this project. I just wanted to point out that the work is available as a preprint. So I would urge you all to look into this work and also use the resource that is available through this project. And I think it would be super useful for researchers who are starting to work on these TREs. And with this, I would like to thank you all for your attention, and I can take questions.
So much, Margaret, for just a wonderful talk, and also for compiling all these really thoughtful resources that seem very accessible and user-friendly. One of the questions I had was, since you partnered with so many of the funders who are setting up the parameters for data sharing as cohorts grow, how did they respond to your insights for the importance of pooled analysis?
Yeah, so they are excited. As you could see, they are there in the work. So they wanted to have a project where in real time we could start and looking into the data and find out the pain points that a researcher would undergo, and then this would help them to change some policies or look into those. So that is the whole aim. So they are open to it actually.
There'll be a reconciliation possible between all of us and UK Biobank for the level of the genome level data.
I'm not sure about it. It all depends at their level, but at least we have, tried to have something to show them,
Great, thank you. Any other questions from the audience?
Sorry, I'm asking too many.
No, that's okay.
Going back to the PC's question is always, I never could understand when is enough. Is PC 20 enough or is PC 25 enough? How did you, what are your thought process? Like, I just want to understand or just ask this question to as many people to understand. When is that enough?
Yeah.
Because that choice impacts the number of variants coming in or falling out.
That is true. Like it also depends upon the cohort, the diversity of the cohort and all of that. But at least for this project where you want to build a workflow, we stuck to a number, that which could be used in both the cohorts. So we have not addressed that question of how many PCs is important. But yeah, I think it also depends upon the population that you're looking at and various other factors.
Thank you again for an excellent talk. And we look forward to your next analysis using this cohort.